Hello friends, welcome back in this video session of medicinal chemistry. So today in this video we will be discussing the drugs acting on autonomic nervous system, which is also referred as the ANS. In this video we will be discussing the introduction of this drugs acting on ANS, adrenergic neurotransmitters, their structures and physicochemical properties, and most importantly the biosynthesis of catecholamines. Adrenergic drugs show their pharmacological and therapeutic effect by either enhancing or reducing the activity of various components of sympathetic division of the ANS. So based on their action we can categorize them into two classes. The first class of adrenergic agents or the drugs which produces effect similar to the stimulation of sympathetic nervous system. So these are called as sympathomimetics or adrenergic stimulant because they are acting similar to the stimulation of the sympathetic nervous system. So they are referred as the sympathomimetics. Mimetics means they are mimicking or they are resembling to the natural sympathetic nervous system. Okay, so they are referred as the adrenergic stimulants. And the other class of the drugs or the adrenergic agents which actually decrease the sympathetic nervous activity. So they are referred as the sympatholytics. Lytics means decrease or removal of this particular activity. So they are referred as the sympatholytics. They are also referred as the anti-adrenergics because they are acting against the adrenergic system, or they are also referred as the adrenergic blockers because they are going to block the adrenergic receptors which are present in the body. So adrenergic drugs or the agents are of two types, sympathomimetics and sympatholytics. Now these adrenergic drugs or the adrenergic agents, they act on the adrenoreceptors which are abbreviated as capital ER small s, that is nothing but the adrenoreceptors, and these adrenoreceptors or they affect the life cycle of adrenergic neurotransmission. Okay, so these are the adrenergic agents which are acting on the adrenoreceptors or they are affecting the life cycle of the adrenergic neurotransmission, and because of this it constitutes a very broad class of agents which are useful in the treatment of various diseases. And because of affecting the life cycle of adrenergic neurotransmission or acting on the adrenoceptors, they modulate many vital functions in the body such as the cardiac contraction, constrictions, dilation of blood vessels and bronchioles, insulin release and many more. So adrenergic neurotransmitters, we'll see what are their structure and their physicochemical properties. So norepinephrine, epinephrine and the dopamine are chemically catecholamines which are generally to all organic compounds that contains a catechol nucleus, which is also referred as the orthodehydrobenzene and an ethylamine group. So you can look at this particular structure, benzene ring which has the hydroxyl group which is adjacent to each other. So this is referred as the catechol nucleus. So this is a catechol nucleus, and this catechol nucleus, when it is attached with this particular beta phenyl ethylamine. Okay, so this beta phenyl ethylamine, so you can see there are two carbons and there is a amino group. So there is a alpha carbon which is alpha to the amino, and then there is a beta carbon which is beta to the amino, which we can also refer it as carbon 1 and carbon 2 to the phenyl ring, or alpha and beta carbon to the amino group. So this beta phenyl amine and the catechol nucleus, the two hydroxy group, if they are present, then it constitute the catecholamines. So this is the structure of the catecholamine. Okay, so this is the ethylamine. So it has a alpha carbon and beta carbon, or carbon number 1 and carbon number 2 with respect to the phenyl ring, and to this phenyl ring if we have two hydroxyl group which are adjacent to each other, which we refer it as a catechol. So catechol nucleus with ethylamine, it constitute the catecholamines. Now the epinephrine are the metabolic products which are obtained from the dopamine. Okay, so norepinephrine and epinephrine, they are nothing but these are the metabolized product which are obtained from the dopamine. Norepinephrine, as you can see in the structure, it contains one primary amino group. So this amino group has two hydrogens, so this is primary in nature. So this is a primary amino group and one primary amino group is present, and there are three hydroxyl group, two hydroxyl group from the catechol nucleus and one hydroxyl group which is actually the beta to this particular position. Okay, so this is the hydroxyl group. So there are three hydroxyl group and one primary amino group which is constituting the structure of norepinephrine, and you can see the numbering of this particular carbons present in the catechol nucleus is 1 dash 2 dash 3 dash 4 dash. So in norepinephrine we can find at 3 dash and 4 dash we have the hydroxyl group, and at beta carbon or at carbon number 1 we have the hydroxyl group which is coming above the plane. Okay, then epinephrine contains one secondary amino group. So if you compare the structure of epinephrine with that of the norepinephrine, you can easily observe now there is a nitrogen which has only one hydrogen. So here it was a primary amino group, now it becomes secondary amino group because only one hydrogen is attached to the nitrogen. So this is a secondary amino group. So one secondary amino group and three hydroxyl group, two from catechol and one at the beta or carbon 1 of this particular chain. Due to this, catecholamine have higher water solubility because there are three hydroxyl group and additionally amino group. So obviously the water solubility will increase, and that's why the log P of these structure or the norepinephrine and epinephrine is in between 0 to 3. Okay, so towards 0 log P indicates the compound is hydrophilic, and towards 5 it will be more lipophilic. Okay, so because of these hydroxyl group and the amino group, it becomes more water soluble, it becomes hydrophilic, and this leads to poor absorption into the CNS. Okay, so that leads poor CNS penetration. Okay, so they will not act on the central nervous system. Norepinephrine and epinephrine both have chiral carbon atoms. So if you look at the structure carefully, so this beta carbon in the norepinephrine as well as in the epinephrine, it has the chiral center, it has a stereo center, because this is a carbon which is attached to four different groups. One, there is a hydroxyl group, other this is a amino chain, and then you have a catechol nucleus, and there is a one hydrogen, okay, which is not shown here but it is present there because of the valency of the carbon. So this is a stereo center, both have one stereo center. So obviously whenever there is a stereo center, we have studied in the organic chemistry, there is a possibility of presence of enantiomeric forms. Okay, so there is a pair of enantiomer exist for non-epinephrine as well as for the epinephrine because of this stereo center. Now in this case, the enantiomer with R configuration, so we have already studied R and S configuration, even we have studied how to determine these configurations, right? Now if this hydroxyl group is beta, okay, it is attached to a beta carbon and this is coming above the plane, so it represents the R configuration. Now R configuration of this particular enantiomer is biosynthesized by the body and it possesses the biological activity. The R configuration of many other adrenergic agents also contribute to their high affinity to the corresponding adrenoreceptors. Okay. Epinephrine drug solutions are added with antioxidants. Why they are added with antioxidants? Because like most phenols, the catechol functional groups are also highly susceptible to the facile oxidation. Now what we do in epinephrine and norepinephrine drug solutions, we usually add the antioxidant to prevent the oxidation of these drugs. So we add antioxidants like ascorbic acid or sodium bisulfite. Otherwise what will happen? The norepinephrine or epinephrine drug solution will undergo the oxidation, and you can see these hydroxyl group are getting oxidized into a keto groups. Okay, so a quinone analog will form from the corresponding epinephrine or norepinephrine, which again undergo the oxidation, and it will result into adrenochrome, and that will lead to the removal of the activity. So to avoid this particular problem, usually we add into these drug solution the antioxidants like ascorbic acid or sodium bisulfite. Now coming to the biosynthesis of catecholamines. Catecholamines in neuroendocrine cells are in a state of constant flux. They are continuously being synthesized and their levels are being maintained. So these are continuously being synthesized, released from this neuroendocrine cell and metabolized, yet they maintain a remarkable constant level in the tissues. Catecholamines are widely distributed in mammals, and their physiological functions are regulated at many sites. The biosynthesis of catecholamines involves sequence of enzymatic reactions. Okay, and these are very much important from the point of designing the adrenergic drugs. So we'll see the biosynthesis of catecholamines. Now it will start with the L-tyrosine, and this L-tyrosine is normally present in the blood circulation and transported actively into the adrenergic neurons. So this L-tyrosine, if you look at the structure, it has a benzene ring, then hydroxyl group which is para to this particular chain and has the amino and carboxylic acid functionality. Now this L-tyrosine, when it goes into the adrenergic neuron, it has the tyrosine hydroxylase enzyme, which is abbreviated as capital TH. Now this tyrosine hydroxylase, as the name indicates, it will do the hydroxylation of this L-tyrosine at 3' position. So you can compare the structure of L-tyrosine with that of the L-dihydroxyphenylalanine, which is also abbreviated as L-DOPA, right? Now here what has happened? There is a hydroxylation at 3' position of this benzene ring or phenyl ring due to the tyrosine hydroxylase. So this tyrosine hydroxylase is hydroxylating the hydroxyl group here at 3' position, and this is actually the rate determining step for the synthesis of epinephrine. Now once this L-DOPA is formed, L-DOPA will be converted to the dopamine with the help of L-aromatic amine decarboxylase, which is also referred as AADC. AADC is nothing but L-aromatic amine decarboxylase enzyme. As the name indicates, it will decarboxylate the L-DOPA. Now you can see there is a carboxylic acid. Now this carboxylic acid will be removed here with the help of this particular enzyme, and it will convert L-DOPA to the dopamine. Now this dopamine will be converted to the norepinephrine with the help of enzyme dopamine beta hydroxylase, which is abbreviated as DBH in capitals. Now what it is going to do? It is going to introduce the hydroxyl group at the beta carbon or carbon number 1 in this particular configuration. Okay, so this is coming above the plane. Okay, it is a beta position. So what it does, by decarboxylation, it forms the dopamine, and then it will introduce the hydroxyl group at the beta carbon, and it forms the norepinephrine. Then norepinephrine will react with phenylethanolamine and methyl transferase, which is abbreviated as PENMET, and then this PENMET will actually transfer the methyl group to this particular amino group. So you can see this nitrogen now get substituted with the methyl group. So N-methyl is transferred here and norepinephrine is converted to the epinephrine. So this is the biosynthesis of catecholamines. It starts with L-tyrosine, then it forms L-dopa, then it forms dopamine, then norepinephrine and epinephrine. So here, as I mentioned, the rate determining step for synthesis of epinephrine is the first step. Okay, tyrosine hydroxylase, which is the enzyme responsible to carry out the hydroxylation of L-tyrosine into L-dopa. So if you have the agents which can inhibit the tyrosine hydroxylase, that will be a very logical or a rational target for developing the inhibitors for these catecholamine synthesis. Okay, so this is with the first part. We have studied the introduction, we have studied the physicochemical properties of these catecholamines, and most importantly the biosynthesis of these catecholamines. In the next video we will discuss the structure activity relationship of these sympathomimetic drugs. Thanks for watching.